Good morning. I am Sombir Singh, Assistant Professor of Botany in Government College for Girls, Chika, Kathal. Today we will discuss about sex determination. These are contents, reproduction, type of reproduction, why sexual reproduction is advantageous over asexual reproduction, sex determination, different sex determining systems, sex determination in human and sex determination in Drosophila and plants. So now we will discuss these contents in detail throughout the lecture. So first of all, what is reproduction? It is a biological process by which organisms give rise to their offspring, or you can say give rise to new organism. So it is the characteristic features of all living organism, the process by which they give rise to their offspring. So why there is a need of reproduction? For the continuity of species. The reproduction is required for the continuity of species. Without reproduction, the species will not able to survive for long time on earth and may soon become extinct. So, reproduction ensure species perpetuation or perpetuation of species on the earth. So now, type of reproduction. Reproduction is of two types, asexual reproduction and sexual reproduction. Asexual reproduction is simpler type of reproduction. It is the reproduction in which formation of sexual organ, meiosis, gamete, fertilization is not required. Only single parent is required and through mitosis offspring are produced. So, the offspring produced by this type of reproduction are exact copy of their parent, and they are called clone of their parent. So, there is no variation. So, it is a simple type of reproduction. In contrary to this, sexual reproduction is advanced type of reproduction. It is the reproduction where formation of sexual organs, meiosis, gamete and fertilization occurs. So it requires two type of reproductive structure. First is male reproductive structure and female reproductive structure. Both these structures are present on different organisms, but in some organisms male reproductive structure and female reproductive, both structures are present. These organisms are called hermaphrodite. Mainly two parents are involved. So next is why sexual reproduction is advantageous. Why sexual reproduction is advantageous over asexual reproduction? Because it creates genetic variation. In case of asexual reproduction, there is no genetic variation. Offsprings are just copy of their parent, or they are called clones. But in case of sexual reproduction, there is a variation. And how this variation is produced? This variation is produced from three different levels. First is allele come from two parents. So in sexual reproduction, two parents are involved. One is male and another is female. Half the chromosome will come from male and half the other will come from female. So genetic variability is there because two parents are involved. And the next level is crossing over. So during meiosis, the parts, or you can say segment of homologous chromosomes are exchanged, and this exchanging of the segment creates new combination of gene, and by creating new combination of genes, variation will be produced. And the third one is reassortment of homologous chromosome. During the meiosis first, at the time of gamete formation, the homologous chromosome are separated, and this separation is independent, or you can say random. We don't know which chromosome will go with which chromosome. So there is independent assortment, and this independent assortment creates variability. This independent assortment creates variability. So here variation is produced from three different levels. First is allele come from two parent, crossing over or independent assortment. So now question arises, why genetic variation is required? Because more the variation is there, more will be the population adapted to the environment. So the next is sex determination. Among most eukaryotes, sexual reproduction consists of two processes. First is meiosis and second is fertilization that lead to alteration of haploid and diploid. First is the meiosis. Meiosis is just the production of just for the production of haploid gamete. So you can say gametes, haploid gametes are produced by the meiosis, and then these haploid gametes, you can say male gamete and female gamete, will fuse by the process of fertilization, and this process generates diploid condition. So there is alteration of haploid and diploid in sexual reproduction. So most of the organisms, sexual reproducing organisms, have two phenotypes. One is male and another is female. You can say sexual phenotype. The difference between male and female is male produces small gamete and female produces large gametes. Mechanism by which sex is determined is called sex determination. In some species, both the sexes are present. Both the sexes, you can say male and female, are present inside the single organism. This is called hermaphroditism or monoecious. Species in which female and male reproductive structure are separate. One organism have female reproductive structure, other organism have male reproductive structure. These are called monoecious. Species in which individual either has male or female, or you say among dioecious species. What do you mean by dioecious? Here male and female are different. Reproductive structure are present in different organisms. Among more dioecious, sex is determined by either by chromosomally, genetically, or environmentally. So these are our next topic is sex determining systems. So these are the systems. First is chromosomal sex determining system. Here sex is determined on the basis of chromosomes. Second is genic sex determining system. Here sex is determined on the basis of genes. And the last one is environmental sex determining system. Here sex is determined on the basis of environment. So now we will discuss this system in detail. The first is chromosomal sex determining system. Hanking in 1891 noticed a peculiar structure in the nuclei of the cell from the male insect, and he called it X body. Later, after this, McClung found that it was a chromosome and he called it accessory chromosome. Now it is called X chromosome. McClung also observed that female grasshopper cells had one more chromosome than male grasshopper cells, and concluded that this chromosome plays a role in sex determination. Now Stephen and Wilson in 1905 found that in insect the cell of female have two chromosomes, whereas the cell of male have single X and another small chromosome. This smaller chromosome is called Y chromosome. It is found in male cells. So you can say Stephen and Wilson found that cell of the female insect have two X chromosome and cells of the male insect have one X chromosome and one smaller chromosome, that is Y chromosome. They also noticed that X and Y chromosomes separates into different cells during the sperm formation. Half of the sperm will receive X, half of the received sperm will receive Y in case of male, but in case of female, all the gametes will receive X chromosome. And they found that sex is determined by this pair of chromosomes, that is XX or XY, and they named these chromosomes sex chromosome, which differ in male and female. In female, they were XX, and in male, they were X and Y. And the other chromosome that they don't have any role in sex determination were called autosomes. So now we will see different chromosomal sex determining system, that is XX and XO, or you can say X naught. Here the organism bearing XX chromosome will be female, and organism having X or you can say X naught will be male. It is found in grasshoppers. And the second system is XX and XY system. Here organism having XX chromosome will be female, and the organism having XY chromosome will be male. It is found in mammals. So this is the diagram showing XX and X naught chromosomal sex determining system. Here XX will be female and X naught will be male. This diagram showing AA. This AA is autosomes plus X and naught. It will be male. AA autosome plus XX will be female. So during the gamete formation, all the gametes that are produced by female will be A plus X. That means female is homogametic. But in case of male, the sperms are of two types, or you can say male produce two different type of sperms. Half of the sperm will have A plus X, and other half will have A plus O. O means not. So if A plus X from male fuses with A plus X from female, then offspring will be female. If A plus naught of the male fuses with A plus X of the male, then offspring will be male. So here the sex of the offspring is determined by the chromosomal makeup of male, not by female. So here the chromosomal makeup of male decides the offspring will be female or male, because female is homogametic and male is heterogametic. So if A plus X fuses with A plus X, then it will be female. If A plus naught fuses with A plus X, then it will be male. So you can say that the sperm coming from male decide the sexual fate of the offspring. This type of chromosomal determining system is found in grasshopper. And the next one that we have discussed earlier is XX and XY. Here XX is female and XY is male. It is found in mammals and also in humans. This is the case of human. 44 autosomes are there plus 2 sex chromosomes are there. So in case of female, there is 44 autosome plus X and X. But in case of male, 44 plus X plus Y. So here also in this system, female is homogametic. It will produce only one type of gamete, that is 22 autosome plus X. But in contrast to this, male is heterogametic. It produces two type of sperms, that is 22 autosome plus X and 22 autosome plus Y. So if 22 plus X gamete from female fuses with 22 plus X from male, then the offspring will be female. But if 22 plus X from female fuses with 22 plus Y from male, then offspring will be male. So here female is homogametic, male is heterogametic. So the sexual fate of offspring is decided by chromosomal makeup of male, or you can say the gamete, the sexual chromosome that is present in a gamete of the male will decide the sexual fate of offspring. If the gamete from male have X chromosome in it, then there will be a female, offspring will be a female. If the gamete have Y chromosome, then offspring will be male. And the next system is ZZ and ZW system. Here, the organism having ZZ chromosome is male, and on the other hand, the organism having ZW is female. So it is found in birds, snakes, butterflies, some amphibians and fishes. And the another one is haplodiploidy system. Haplodiploidy system means haploid and diploid. Individual having haploid condition will be a different sex. Individual having diploid condition will be a different sex. So haploid set is there if in any organism, then it will be a male. If diploid set is there, then it will be a female. It is found in bee, wasp and ants. So this is the diagram showing ZZ and ZW chromosomal sex determining system. So here ZWA plus A is showing autosomes plus ZW is a female, A plus A autosome plus ZZ is a male. So here the condition is reverse. Here you can see the other system that we have studied, that is X naught and XX, or you can say XX and XY. In both of them, female was homogametic and male was heterogametic. But in case of this system, female is heterogametic, that is ZW, and male is homogametic, that is ZZ. So here male produce only one type of gamete, that is autosome plus Z, one set of autosome plus Z. But in case of female, it will produce two type of gamete. Half the gamete will be A plus W and half will be A plus Z. So if A plus Z from female fuses with A plus Z from male, then offspring will be male. But if A plus W fuses with A plus W from female fuses with A plus Z from male, then the offspring will be female. So here female decides the sex of offspring, or you can say the gamete from female decide the sex of offspring. If the gamete is A plus W, then offspring will be female. If it is A plus Z from female, then offspring will be male. So here female is heterogametic, male is homogametic. And the next one is haplodiploidy sex determination. So this sex determination is determined by number of chromosomal set. So you can see from diagram, in parental generation, female is diploid, male is haploid. So if female is diploid, then during gamete formation there will be a meiosis, and meiosis will reduce the chromosome set from diploid to haploid. So gametes will be haploid from female, and male already is haploid. If gametes are formed, the gametes will be formed from mitosis, and gamete will be also be haploid. If gamete from female and gamete from male fuses through the fertilization, then diploid condition arises, that is 2n zygote. So if diploid condition arises, then offspring will be female. If the gamete from female is not fertilized, it remains haploid, and this haploidy give rise to maleness. So any egg that is fertilized will become female, and any egg that remains unfertilized will become male. So here number of chromosomal set determines the sex. So we have discussed four different chromosomal sex determining system, that is XX and X naught. Next was XX and XY, and the next one was ZZ and ZW, and the final one was haplodiploidy. Now genic sex determining system. In some plants and protozoans, sex is determined genetically. So there is no difference in the chromosome of male and female. There are no sex chromosomes. The sex is determined by presence of genes. Genotype at one or more loci determine the sex of individual. It is important to understand that even in sex chromosome determining system, sex is determined by genes that are present in the sex chromosomes. For example, SRY gene located on the Y chromosome determines the maleness. In both chromosomal sex determining system and genic sex determining system, sex is controlled by genes. Then what is the difference between chromosomal sex determining system and genic sex determining system? The only difference is, in case of chromosomal sex determining system, genes are present on specific chromosome. For example, in case of XX and XY, all the genes that influence the sex will be on X and Y, not on autosomes, not on other chromosomes. These chromosomes have all the genes that influences sex. But in case of genic sex determining system, there is no such distinction. The chromosome gene may be present randomly on any chromosome. They are not specific to any one or two chromosomes. They may be present in any chromosome. So here, chromosome of the organism in genic sex determining system, unlike chromosomal sex determining system, will be similar of male and female. There is no distinction in chromosomes in male and female, but in case of chromosomal sex determining system like XX and XY, there is a difference. Female is XX, male is XY. So this is the difference. Next is environmental sex determining system. In Rhapidula fornicata, Sliper limpet, this is the ideal example of sex determining system. This organism lives in stack. This organism begins its life as a swimming larva, and the larva settle into solid substrate or unoccupied solid substrate, and develops into female lymphet. After developing into female lymphet, it will secrete some attractants, some chemicals that will attract the other surrounding lymphets, and other larvae then attract towards it and settle on the top of it, and the larvae that are on the top of this limpet develops into male, and they serve as the mate for the limpet below. After a period of time, male on the top develops into female, and they attract other larvae, and the other larvae then settle on the top of stack, and they develop into male and serve as the mate for limpet below. Limpet can form the stack, dozen of stack. The uppermost larvae always will be male. So this type of sexual development is called sequential hermaphroditism. Here, individual can be male or female, but one at a time. So, in case of Rhapidula fornicata, sex is determined by environment, by the lymphets position in the stack. Here, sex is only determined by lymphet position in the stack. If it is below, then it will be a female. If it is on the upper side, then it will be a male. So, the first larva that will occupy on any unoccupied substrate will be a female. It will attract others, and the other larvae that will gather above this female limpet will be a male, and after some time they develops into female, and then in turn they also attract other larvae will come there on the stack, and they will develop into male. After that they will develop into female and attract others. So here the sex is determined by position of organism in the stack. If it is on lower position, it is below, then it will be female. If it is above, then it will be male. So the next is sex determination in humans. We have already discussed it in our part, that is chromosomal sex determining system, XX and XY sex determining system. Female is XX, male is XY. Female is here homogametic, male is heterogametic. And the next one is dosage compensation. Dosage means here dose, compensation, to compensate the dose. So you can see from this diagram here, female have 2 X chromosome and male have only X, and the other is Y. Y is a short chromosome, it have fewer genes, only few genes are there in Y chromosome. So the dose of the gene is double in female than male. So to balance this dose, there is a mechanism in females. So we will discuss this mechanism. So you can see male are heterogametic, female are homogametic. So female have 2 X chromosome, male have 1 X chromosome, and the other Y chromosome, that male have only fewer genes are there, and it is small chromosome. So normally female must have double dose of genes that are present on X chromosome, but it is not, because the 1X chromosome is in female is inactivated early in embryonic development. This inactivated chromosome is called bar body. It can be seen as highly condensed region inside the nuclear membrane. Highly condensed region inside the nuclear membrane is called bar body. So this inactivated X chromosome is called bar body. It is found only in females because male have only X chromosome. So it is for compensation of dose. So if any organism, any male, any human who have X, X, X and Y, then except one, all the two X will be inactivated. So you can say only one X chromosome will be activated for the dosage compensation. And the next one is sex determination in Drosophila. So in Drosophila have 8 chromosomes, 3 pairs of autosomes, means 6 chromosomes are autosomes and 1 pair or 2 chromosome are sex chromosome. Female normally have 2X chromosome and male have X and Y chromosome. Presence of Y chromosome here, it is very important. Here Y chromosome does not determine maleness. In case of Drosophila, in case of humans, Y chromosome determines the maleness, but in case of Drosophila, Y chromosome does not determine the maleness. Instead, sex is determined by balance between genes on autosome and genes on X chromosome. This type of sex determination is called genic balance system. Here a number of genes seems to influence the sex development. The X chromosome contains genes with female producing effect and autosome contains the genes with male producing effect. So simply you can see that in very simple language, sex is determined by X and autosome ratio, X chromosome and autosome ratio. The number of X chromosome is divided by haploid set of autosome. So you can see from this table, if any Drosophila have XX and haploid set is AA, then ratio is 1. If the ratio is 1, then Drosophila will be female. If XY AA, then ratio is 0.5, then Drosophila will be male. And the third one, X naught and AA, the ratio is 0.5, Drosophila will be male. Here important to note is XY is not influencing the sex. If Y is there and in case of third Y is absent, in both the cases Drosophila is male. So Y chromosome does not determines maleness. If XX and XX and Y, then ratio is 1 female, and if ratio is above 1, then whole will be meta female. If ratio is above 0.5, then the organism will be meta male. And the last one is sex determination in plants. Here we have taken only one example from Melandrium album, that is XX and XY type. It is the most studied plant, dioecious plant. So in Melandrium album, the sex determining system is XX and XY. Here female is XX, male is XY. So female here is homogametic, male is heterogametic. So the gametes from male will decide the fate of offspring. And at the end we have some multiple choice questions. The first one is the chromosome responsible for determination of sex are called. Second is in human females are hologametic, homogametic, hetero, hypo. In humans, the chromosomal condition of 44 autosome plus XX refers to. Which of the following type of sex determination is found in birds? Number of autosome in man. In case of human being, chromosomal makeup of which sex will decide the sex of offspring? Which chromosome have no role in sex determination of in Drosophila? In Drosophila, sex is determined by the ratio. And the next one is genetic balance theory uh, of sex determination was given by Bar body are found in humans and are associated with. So these are MCQs. Thanks.